changing, and we are honored to have two very special guests from Regeneron, which to me is an iconic company in the modern generation of biotechnology because of its unique drug discovery platform for monoclonal antibodies, which has led to a wonderful collection of, I think, 7 commercial drug products that are out there now. And it is right here in New York. Who we have here is Hannah Bach, Senior Vice President of Preclinical Manufacturing and Process Development. So just in case anyone wonders what that is, Hannah has led roles in purification development and also process development. When you're doing process development, you need to do analyticals. So also head of analytical support for that. Also preclinical manufacturing, and that's a smaller scale. So when it goes to a large scale manufacturing, technology transfer of that to many manufacturing sites. I was surprised when I saw that Regeneron not only has its own 11 manufacturing sites, but also sites that are probably contracted around the world. And so this is a vital link between R&D and manufacturing. And also in the regulatory filings, her team has provided plenty of support for that. Hannah has a PhD in chemical engineering from the Technical University of Denmark. And we also have Barry Cole, Vice President and Head of Global Clinical Operations. That may sound like a very involved topic, and it is, because that's where a lot of the cost and time is consumed in drug development. Clinical operations involves clinical trial management, site manufacturing, site monitoring. You have clinical trial monitors and management of that process and the process, global trial optimization, because these are very expensive activities, and all the systems behind that, study planning, obviously training, SOPs, standard operating practices and processes. And therefore, when you do all of that, you are also in the therapeutic area, project management, clinical logistics, and the vendor management, which involves business operations. So also not so technical, but also probably on the finance side. Barry also was Vice President of Business Operations Analytics at Pfizer, as well as Strategic Alliance and Development Operations. And prior to that, Vice President of Clinical Operations and Strategic Alliance Management at ICON, which is a CRO, contract research organization doing work in clinical studies. And Barry has a master's in neuroscience from NYU. So let's just get right into it. Our audience, I presume, and please feel free to add your questions into the chat. And I presume many of these in our audience now are in an academic setting or in a startup setting. And it used to be that from the tech transfer perspective, one could have a molecule, large or small, or an assay, and in a tech transfer office, just license that out to a pharmaceutical or biotechnology company. Maybe, yeah, that might have happened decades ago, but that's not so much happening anymore, because major companies would actually like to spend more money and actually acquire a company that's actually got a product that's either approved or where it's mostly de-risked, at least to the perspective of the company that's doing that. So the need for any developer or inventor to get something along would have to be, they would have to be much more involved, perhaps get a team and actually start a company or do something to move things along. So my first question, I'll actually ask both of you this question, is that if one is in an academic setting, we often see that there's a path to IND, investigational new drug application. And they're trying to raise funds and do specific steps to that. And I'll ask you, each of you, what is that? And from your perspective, what do you look at when you actually want to look at a molecule or some project plan getting towards that? How about I start with you, Barry? I just see you at the top of my screen, so I'll do that.
Actually, I just to be chronological, Hannah's group usually comes before mine in the drug development process because of the need to go from the research and discovery space through to the development space. And so I'll turn it over to Hannah to really start off where the process starts, and I'll then fill in once she gets to where she would hand off to groups that I'm working with.
Right.
Great.
Thank you, Barry. So I think before I answer the question or seek to answer the question, I think it's important to highlight that Regeneron is a unique company in that sense, as well as many others, and that is that our pipeline is entirely homegrown. We have never acquired a molecule from anyone ever. So every single molecule we have in our pipeline has been developed in our research labs, in our discovery efforts, and taken all the way through to whichever stage they're at now. The outlier there, of course, would be we have in recent years made a number of strategic alliances with collaborators where you could argue that that's a version of buying a molecule, but maybe different from the thought experiment or the question you had, Sam, around buying it directly from an academic institution, because that is not something that we've ever done. But when we have looked at strategic alliances with other companies, it has very much been about, can we find a company where the science is sound and where their skill sets are complementary to ours, so we through that can find synergies and maybe even combine their technology with our technology, and through that do something novel. So we're not just acquiring something that's an add-on, but we really want to make something with it and find synergies. So I know that doesn't really answer your question, but it's because we've never done it. That said, should we do it? It would again be going back to maybe if I can take a step back. The way I like to look at drug development is there are really three key questions you need to answer. The first question is, does it work? And you don't know that till you've done it, but you need to interrogate that very closely in both in vivo and in vitro experiments preclinically, and then of course clinically later on. The second question is, can you make it? Can you make enough drug to get into your first animal study? And then can you make enough drug to get into your first human study? And can you make enough drug to supply the market? I think a famous example there is with the Ebola outbreak in East Africa, where the ZMapp molecule, which was a 3 antibody cocktail, they were made in tobacco plants. So as that epidemic was raging, I think the total world supply was 6 doses. And it's hard to make a clinical trial or even have a commercial drug with 6 doses. So that was an example of, can you make it, was not an absolute yes at that time. And of course, the third question is it safe? And that is, you can determine through understanding the molecular profile and having an experiential base, especially if it's a monoclonal antibody, you know what kind of good looks like. And then, of course, it's interrogated in the clinical trial. So just to step back, does it work? Can you make it? Is it safe? I think are the 3 key questions. And so if you look at it through that lens, that would be probably the first lens you would look at if you look at a molecule in an academic institution, is what is the probability of success for those 3 questions? That would be where I would start. Bari, I don't know if you have anything to add.
That's great. And we didn't plan this, but I can talk about the 3 questions I would ask that come after those early questions are asked, because when we're working collaboratively between research, discovery, and development, we look early at a few different things. Some of them are related to some of the aspects of safety that Hanna was talking about, but also what is the current treatment landscape? So if we have a good molecule, we believe it'll be safe, we think it's going to work, we need to make sure that it can fit within the treatment landscape that exists out in the world and in the paradigms that exist at treating institutions and organizations. And we also look at protocol. Can we design a protocol that will answer the right questions? And what would we need to do that? And what would that look like? Can we then operationalize it? That's where my group comes in, working very closely with the physicians and clinical scientists to really look at what would that design be? What would the regulators need to see when you're filing an IND in terms of your analyses? And also the endpoints that you're going to look at to not only show early on in early phases, that you have safety, but then as you go further, that there could be some potential efficacy. From an operational lens, once we've passed all those hurdles, we actually have to make sure there's the population actually that we can treat and that there's access to. And so there's other considerations around even competition and trials that are out there and the ways in which we can study the right population. So there's probably a host of other areas to look at, but I would say those are the key things we look at in terms of protocol design, the treatment landscape, and the ability for us to actually operationalize and carry out a study that's going to give meaningful data.
Thank you. So let's look at some things. I'll just ask a specific example. I remember I was at a session once hosted by someone at FDA, and what they were highlighting was that in the immuno-oncology space, which is very hot and still is, there's all these investigator-led studies, and these are just some PI who has got access to some patients, and they were looking at the CMC part of the FDA was looking at the chemistry, manufacturing, controls direction of this, and just without naming names, put some examples. They ordered some peptides from a supply house, which is not GMP. They mixed it with others, and no consideration for formulation, and this was going to be injected and into some patients, sterile filtered, of course. So that did not fly with FDA. Many compounds nowadays are fairly complex, not just in just the active ingredient itself, but the formulation. When you've got work like that to do, plus you've got animal studies and human studies, and you've got limited costs, what advice can you give to a team that's strapped for cash? At what point should they really think about GMP-like or GMP manufacturing or some direction of that, while at the same time trying to save on the costs?
It may sound too simple, but I think you want to make sure you have a molecule that works. And I know, of course, you don't know that, but really be very critical about your data and your hypothesis. Did it really work in your in vitro experiments? Did it really work in your animal models? Do you know whether your animal model is predictive? How did it do in the monkey tox study? Do you have the right dose, et cetera, et cetera, et cetera? And until you can unequivocally say yes to those studies, if you're cash strapped, you may want to hold off on GMP manufacturing, which is, a big step up in the cash you need to put out, especially if you go to a CMO. And at the same time, it's a push-pull, because now suddenly your CMC is critical path, and it's hard to wait. If you feel that you have a compound and then you go to a CMO and they tell you, yeah, we can do this for you in 18 months, that's a long time to wait. So, but that's really the optimization you're looking at. How long can you wait till you pull the trigger on GMP manufacturing versus how sure are you that you're really ready for clinical trials? Because you need your GMP material before you can file your IND, and you can't dose any patients till you have your IND, right? Except maybe investor-led studies, and Barry, you should comment on that there may be some single patient INDs or other opportunities, but I don't know enough about that.
Yeah, I'll just say we do support at Regeneron a whole host of investigator-initiated studies where if they have a good protocol and a good idea, we will support that with some of the molecules that we have in early testing. But I from taking from what Hannah said, I do believe that the timelines are critical, and it is an impeccable planning that's required in the early stage to really ensure once you believe you have the data that's going to be meaningful and enable an IND, that you have the rest planned in parallel, because ultimately once you file that, you need to be able to have the drug availability to then support the types of sites that you need, the numbers of patients that you need to dose, and an overage in addition to ensure you cover a longer time frame and expiry and other things that we need to consider throughout the process. Things don't always go as planned. And even when you would like to have your IND and you expect an IND date, you may have questions that come out of that. And that could be another 6 months that add to the timeline in going back and forth with the FDA or other agencies. So these are all things that a lot of things have to happen in parallel, but you want to have some certainty because there's a lot of effort that goes into that.
Right. Okay.
And maybe to add to that, Barry, it's really surprising how many things can go wrong. So overage, yes, absolutely. The number of stories we can tell about, trucks not arriving and things thawing out because they got stuck in customs. It's more often than you would think. So again, it goes into the planning. We don't do anything for clinical trials, but we do ship material out for IND-enabling monkey tox studies. We never ship on Thursdays, ever. Even though it's an overnight FedEx, we never ship on Thursdays. It has to be Monday or Tuesday preferred, Wednesday maybe, because FedEx overnight is not always overnight, and now you end up on a loading dock somewhere, and nobody looks at it till Monday morning, and the dry ice is gone. So those are some of the things that also go into some of the detailed planning. It sounds very trivial, but your material is gone.
That's very interesting. So just for the audience, I don't know how much the audience knows about this. So in order to have an IND filed, one needs obviously the in vitro data, but then you need in vivo animal data to prove that it actually works. Plus,
that it's safe.
It actually works in the animal. And then the question is how appropriate is that animal model for the condition you are trying to treat. And then there are also a series of safety studies that it turns out to be more expensive as you start to get to more complex animals and larger doses. So these are all very expensive items that need to be checked off in a project plan before it actually gets into human studies. And that's the challenge of actually getting material and also making sure that all your data is appropriate to justify that this is something worth going into humans. Now, once you do go to humans, then the question is, for, again, a company out there who is resource strapped and probably are more on the science side, so hasn't thought about who to give this drug to and how you're going to do it, one would actually need to find a lead investigator or someone or would that be how would one, Barry, what can you tell us about how you would do that? Perhaps
It usually helps to have an advisory panel or a lead investigator, some key opinion leader in the area to really support the types of studies that you're going to do. And certainly though, we don't always do that. We get that background and that feedback for the program planning, if in fact we believe there's some potential for a molecule early on. But in your first in human study, we typically do our studies in phase 1 clinics. They're very well staffed and organized and planned and get normal healthy volunteers. Where it's a little more challenging is in the immuno-oncology space or areas where you don't go into normal healthy volunteers first. And there you actually do start with a lead investigator and what you would do in later phases in other areas. So it really depends on the therapeutic area that you're going into. But for the most part, your first in humans are in early phase 1 units to really determine the PD/PK profile of the molecule and get some early safety signal. But we at Regeneron have a large immuno-oncology profile. And so in many cases, we're already going to academic institutions and multiple lead investigators to really spearhead our efforts and align us with the community, because ultimately the treatment paradigms, especially oncology, change month to month at some points in time. And when new data comes out, that could shift the design of your study. So we do try to be tightly aligned with those in the medical community that are treating patients in the area that we're studying.
So you touched on the question that you gave an answer that I was going to ask about lead investigator or lead investigators. How many, and how would you make that decision? Why would you want more than one, or how many would you want? What's the thinking behind that?
It depends. Again, it's very therapeutically aligned, because in some areas you may, and depending on where you would be looking to do the studies that you're doing, where you're looking to ultimately market the drug potentially, but the various medical communities globally do see different molecules differently and allow for testing in some areas that others don't. There are particular standards of care in different regions. So there, in particular, early on, you may be working with one lead investigator because you may be doing your earliest study, let's say in the US or in the Netherlands. But going further than that, you may have a drug where you actually have to start testing it in multiple locations in rare disease, for instance, where you need multiple investigators because the paradigms are different based on where you go. So that would essentially drive some of the thinking on lead investigators. And some countries actually require you to name a lead investigator in order to do work in that country.
Mm-hmm. Let's look at where things are going now, today, versus years ago when you were working in this area, about when plans get locked in. So, and I'll ask this both on the clinical side, clinical study side, as well as on the manufacturing side, because once you get into human studies, things change. Maybe the formulation has to change, or you've discovered something. How is that changing the process, or how much rigor around that, or if it's not the rigor, it's methodology that FDA may be changing these days, and what's the state of the practice that you've been doing to make sure that you are compliant, but at the same time adjust for things that are happening?
Maybe I can start. I don't, at least, the majority of our portfolio is monoclonal antibodies and our own version of bispecific. So, and once they are produced and purified, in many ways, they are similar to a monoclonal antibody. And for those molecules, I wouldn't say things have changed that much from a regulatory perspective with regards to what you need to do if things change. As you mentioned, things change, maybe your formulation change, maybe you want to go to a different manufacturing scale, maybe you started your clinical trials with a, let's call it first in human manufacturing process, and you now want to have a manufacturing process that could supply commercial demand. So there will typically be a second version of the manufacturing process. And in all instances, whenever there is a manufacturing change or a site change or formulation change or anything changing in the CMC space, it is the obligation of the sponsor to demonstrate to first themselves and then the FDA that the drug will have the same efficacy, potency, and safety as the one used in previous trials. And that's a comparability package. It's described in ICH Q5E. And depending on the change, it may be sufficient to characterize your product. But in many cases, there will be, you will need to do clinical bridging to illustrate to yourself and then the regulators that the version 2 of the drug is the same as the version 1 of the drug, such that you can leverage the prior safety and or efficacy data you have on the drug. So I don't think it's changed a whole lot, because that's been the case all the time, and the guidance is I think more than 15 years old.
Great. It's good to know that what's working is always continues to work. You mentioned something about clinical bridging, but bridging in general is an interesting question, because sometimes one may have a supplier of a product, And then for a backup, you may want another supplier, but the the manufacturing processes are not going to be the same. And if it's a complex product, then the product will not be exactly the same either. There will be some bridging or perhaps not bridging, but something to be done to make sure that both of those suppliers are at least suitable for the drug product or drug substance. And so the question is, do you have any sense of how much effort, how much money, or what one should do to, how much money is involved to do that, or what should one do to make sure that the product meets whatever specifications you set?
Right. It's a lot of work, which is why process changes and site changes or CMO changes, I think, should be done thoughtfully and with intentionality. And the kind of things you do will depend on your target product profile. So it's a matter of, you need to understand your molecule well enough to understand what quality attribute could potentially change efficacy, safety, and potency. Because even in ICH-Q5E, it is described that there can be analytical changes. But again, you need to look at them and risk assess. Is this a change that we, based on our understanding of the disease state and the molecule, will have a meaningful impact? If there's a potential that it has a meaningful impact, it will be required that you do clinical bridging. So to maybe turn the question around, Sam, and say, rather than say, well, when I go to CMO 1 and CMO 2 and the processes are different, I would say work with your CMO to make sure they're not different, because that's going to save you a lot of time and effort and money. And if you understand your manufacturing process well enough, that is possible.
Great advice. Barry, over to you on the clinical side. And I think this might be perhaps, I don't know, maybe more interesting, because at some point in my life, I heard about phase 0 studies, for example, and now there's maybe other things happening. What can you tell us about how clinical studies are being done more effectively or more efficiently?
So there's a couple things, and I think Regeneron, given our foundation in science, is really good at determining the potential for our molecules early on. So we do what we call experimental science studies, and we've done a host of, whether it's observational studies to look at populations, we've done studies to look particularly at biology or imaging practices, we've screened patients for particular biomarkers to look at the incidence of the population. So we've done we do a whole host of effort prior to even getting into a clinical trial to better design our protocols and really look at, can we design the right trial given what we know about the molecule that will have a better chance at what we know in terms of the profile of our product. And I think in terms of other changes, I will say clinical development is changing, but it's been overall pretty static for years. And what we're seeing now is a digital evolution. I think with data, both the genetics data, Regeneron has a genetics center where we're working with many institutions around the world to bank genetics data. A lot of our efforts are also supported by actually looking early on at some genetics data that'll give us some signals to molecules we should be looking at and populations we should be testing, and how we think they'll react to the products that we're creating. And so I think that, to me, is the evolution of development, is really in the supporting data that's out there that hasn't really been, we may have had some of this over the last, I'd say 10 years, but we haven't actually mined the data to the degree that we could, using some of the technologies that's out there to really look at how we can develop drugs better. And a case in point, we work with a consortium of all the large pharma and biotech companies. And they actually have collected from all of our companies, Regeneron, Pfizer, Abbott, Lilly, all of the big companies, placebo data. And so we're sharing data now, not only across companies, but throughout the industry, to try and look at response to different types of therapeutics to really utilize that data to plan trials. And we've done that recently with COVID as well. And so I think these are the things that are changing drug development, is really the ability for us to look earlier on at data and make some assessments that'll help us better decide what we want to look into, but also decide what the design of our trials will look like.
Who would you, I presume you need some genetics experts of some sort. What new skill sets do you need to have to enable this?
So it's interesting because over the last, I'd say, 10 years, the Genetics Center has continued to grow and add people with obviously a background in genetics, but also data science. I think there, and technology. I think you need people who are systems experts. We've, a lot of our sequencing required robotic automated processing and robotic automation. So people who have experience in technology and innovation, people who are data scientists and can look at the data and really pour through and figure out how that might work. AI is coming into play as we look at imaging data. So it's really these skill sets that, I do believe there have been people in our industry and even at Regeneron who have some of them, but we've actually been, even in my group, I have a data science group that looks at really connecting all the disparate data from our clinical trials, because we utilize many vendors, whether it's a lab, a specialty lab, an imaging vendor, we're now doing a lot of wearables. So getting the data from those wearables that people are using in their daily lives and incorporating that, those don't go into a system that we've set up. They're in the ether, they're in the cloud. And so having the ability to mine that data as well and bring it all together and make it talk to the other data that we have is a challenge. And those skill sets are incredibly important these days. I'm hiring more and more for people who have that type of analytical mindset.
We have a question from the audience that I don't know. I don't know enough about antibodies, so maybe this might not apply to you, but you may be aware of situations. What if the mechanism of action is not really known? I know there are drugs like that. How would you evaluate, or what would you, is it always more the case that the mechanism of action now is known nowadays, particularly if you deal with antibodies, or maybe perhaps not if it was an immuno pathway?
I would hope you would at least have a hypothesis that you're able to test. I know that sometimes for small molecules, there may be less of a mechanistic understanding. Maybe the question links back to understanding the quality attributes and doing a risk assessment, right? The simpler your molecule, the easier it may be to characterize it sufficiently to understand whether there is a difference or not. With monoclonal antibodies or big proteins or Fc fusion proteins, it can be a little harder to do so. So the more complex your molecule, I would imagine, the better you need to understand your mechanism of action in order to be able to engage the FDA in those discussions. And I would say, when in doubt, the FDA will ask you to do more. So the burden on proof of proof is on you as a sponsor. And if you can't prove that product A and B does not affect efficacy, safety, or potency, they're going to ask you to do more clinical trials or maybe go back to safety studies. So that burden of proof is really on you as the sponsor.
There are some companies going towards more orphan indications and rare diseases, and so those are situations where you're not going to have a huge recruitment of patients. And if the mechanism is somewhat known, perhaps genetically or through the mechanism, and there are perhaps a few models about for that condition, and if it's reasonably consistent across those models, would that be sufficient to run a smaller trial, or how would one go about that?
I think it's a really good question, and I think you've laid out the what you need to think about. Is there enough of a population, and have you have your early test really confirm the target and confirm the mechanism. And if you truly believe that it will work, ultimately it's a risk no matter what you do. I think you have to ensure you also have designed the right study. I think that's often, the biggest risk you take is how you design that study. Once you have a really good signal in the preclinical work that you've done, and you can confirm the mechanism, at least to the degree that you can before your clinical human trials. But ultimately, you don't have a large chance with a large population is what it comes down to. And you do have to really figure out the right procedures to include, the right labs, imaging, biomarkers, and ways in which you want to look at the outputs. Because I do think one of the things we do is we try to think about all the things that we might want to look at in the trial and the things that might give us a signal downstream. And it's not always that those that we thought would be our primary endpoint, and I think all drug developers find this, turn out to be that. But if you really have a drug that works, good drugs aren't that subtle. And so you can look at some of your data and really determine why it worked and what about it worked and how can you show some efficacy.
Let's turn our attention to COVID right now, because I think that's a topic that's on a lot of people's minds. I'm sure things are changing. I'll just ask one that I'm aware of or I think I'm aware of, is that it's harder to run clinical trials because patients are not showing up or because of various issues with social distancing and so on. How's that going? How does one go about doing that now? And
it is tough. It is tough. I will tell you that there's a silver lining, though, I think, in the industry with regard to the way the trials have to evolve because of COVID. Because I do think that some of the things that we do today or are required to do, we need to rethink. But it is challenging. We have not in the past shipped drug directly to patients. We have put in place procedures to do that, where patients actually are able to do self-injections. We have usually they go to our clinic, whatever site we're running this study at, they're given drug and that's essentially how it's dispensed. In this new environment, we ship directly to patients where we can. That's very different in the world of clinical trials. That is typically not what sponsors of studies do, but that is something that's on the docket to have discussions with the FDA and others about making a pretty standard process if we need to. And even for trials to think more about patient-centric trials where patients don't always need to go to a clinic to do some of this. So I think there's some positives that are coming out of it, but for today, we are, in most countries able to run trials. We have progressed the majority of our pipeline. There have been sites who have not been able to see patients, but as the environment has evolved, they've also gotten back on track with the ability to open slowly, put the precautions in. We've changed some of the consenting that we've done. There's a lot of hospitals requiring COVID tests now that didn't before. It's really adapting to the environment. I think the way many businesses we have as well, and as have hospitals and clinics that are testing patients. So it's a challenge, and we've spent a few months trying to figure out how to manage that challenge. But I would say, as of today, while things aren't back to normal in clinical trials, we're well on the path, and we are, as I said, moving the majority of the portfolio along.
Okay, that's interesting. I won't ask you about Regeneron specifically, but industry-wide, what's your sense? Is this slowing down at how many months, the duration of, say, pivotal studies? Yeah,
I sit in a lot of leadership meetings across operations leaders from a lot of the companies that are out there. And I don't know that we've specifically talked about the months of delays that we might incur, but I would imagine that, depending on the population that you're treating and the importance and criticality of some of the drugs out there or that are being tested in trials, there likely will be some effect and some delay to what we would have seen in 2020 versus what we'll now see going into 2021. So I would say that every trial that's out there has some potential for delay, but it's really how you can, trim that down. And if you have a well-run trial and you open up additional sites, you can actually, overcome some of that. And so that's what really companies are doing now, is trying to backtrack and figure out, how can we cut time on the back end? Can we open this up to more patients? And I do think the FDA and other agencies are also working with us to look at our sample sizes, to really figure out, are we able to look at a slightly smaller sample size than we originally intended? What are the implications to doing that? So I do think it's not just Regeneron or any companies, it's actually the whole industry is really trying to look at, how do we not slow down progress while we're in this state, and at the same time, ensure that there's a real focus on COVID and finding a treatment for COVID as well.
Hannah, what's going on the manufacturing side, as well as everything else that you've experienced? Any new innovations on how to solve any issues that arise?
Well, I think the short answer is yes. I mean, I think we try to innovate constantly in what you do. And I'm looking for a question behind your question, whether you're speaking about linking it to COVID or in general.
Oh, yeah, COVID in general, because I don't know if people, with social distancing, I don't know what happens in a facility, but I presume that people are masked anyway, or
yes.
And then there's the supply chain issue.
Absolutely. Yes. So, at Regeneron, and I, as I imagine many other places, one of our primary focuses when the pandemic was really at its peak in New York State, where we started out being the epicenter for the pandemic in the U.S. It was a real focus for us as an organization to keep our manufacturing plants safe. And by safe, it means we were very, very focused on making sure that the people that work on them in the manufacturing site, that they were safe from infections, such that we could keep making drug. Because if we can't make drug, we can't supply our patients, right? So we have existing drugs, we have existing patients. So we wanted to make sure that we could keep helping the patients that are benefiting from our drugs. And so because of that, we had some very strict travel restrictions. So under normal circumstances, my team as a process development organization, we would typically travel to the manufacturing site when we implement new manufacturing processes, and collaborate with our colleagues there on making sure the process runs as intended. But we basically shut down between site travel because we didn't want people to travel from New York City and Westchester up to Albany, where the manufacturing site is, because there was a lot less disease spread up there, and we didn't want to unintentionally contribute to making that worse. So we had to figure out how to do technology transfers virtually, which we had never done before. And that has required some innovation. We've also had to onboard new employees. They need to get trained in how to execute activities in the lab. So we've been testing out virtual headsets, such that if somebody who's very skilled in a specialized activity such as robotics or automatic liquid handlers, if they for whatever reason could not or would not work on site for personal reasons, they would still be able to train people in running things in the lab through using virtual headsets. And through that, we've been able to keep a lot of our operations running, both on the R&D side, process development, as well as manufacturing, through having a lot of intentionality about who's on site, what are the safety measures, how are we protecting ourselves as well as the site. And that's been quite the journey. But I think we're very happy to share that we've been able to do that with no community spread at all.
Yeah, I saw just a year ago, before all of this happened, a local company had Google Glass was one of them, but there were others and it was particularly adapted for or developed for pharmaceutical manufacturing, as well as a lab work. Do you use those at all?
Well, we do have something that's we're testing out something that I would say is similar to Google Glasses. It is this virtual headset where you can hear the trainer in your ear, and there's a microphone, and then there's a little camera that allows the trainer to see what the operator is seeing, and that has enabled us to do some of the work that we otherwise would not be able to do. So that's been one of the tools that we've tested out with some good success.
We have a question here from the audience that is very specific, but I'll ask it just because we want to serve our very curious listeners out there. And this with regard to drug delivery. And what if the device, or I don't know if it's a device or just a different mechanism of delivery, is affecting the concentration of the API? Would that require a different clinical trial? That's about the question that I got.
If the device affects the concentration, why would it do that?
Yeah, how do delivery mechanisms affect the need for clinical trials? If a new delivery mechanism decreases the API concentration, so it might not be a device, maybe it's a delivery mechanism. So maybe it's a drug delivery technology.
Well, if it's a drug delivery technology, I'm speculating here, it sounds like there's a question behind the question. If it's a drug delivery technology that changes the concentration, my assumption here would be that we're talking about a sub-Q administration or intramuscular administration, and then you want to make sure that your bioavailability is unchanged. So I would assume that that would require some sort of clinical bridging to make sure that the old and the new delivery system gives equivalent bioavailability of your drug.
Yeah, I would agree. And we do, when we run trials early on, very often we're running trials in IV, and then we switch to sub-Q, and we often do smaller bridging studies to ensure that. And in addition, I mean, even when we've changed from sub-Q and potentially used, went from a vial to a pre-filled syringe, we've also done small studies to ensure that the delivery device is actually providing the same concentration and bioavailability.
Right. I love that answer.
Right? Because often when we've gone from a vial to a pre-filled syringe, it will often be the same concentration. So it's really just a container that has changed, correct?
It is.
Yes.
It is, but often, the container does change, but it's a matter of, when they draw it up and when it's the operator. At that point, it becomes more the operator than anything. But if you can show the same results, then that's usually enough to persuade the agency.
Yeah, I love both of your answers because it really exemplifies how you're thinking through the question and where the issue might lie and how you would address that, which is science and technology and operation. Let's get. I want to ask a personal question here because FDA hasn't changed the drug a lot, as we've heard, and this is the practice of the industry for a while now. When you first came into this industry, you like all of us, probably starry-eyed and excited. Are you still like that? What keeps you excited now? And what do you really want to pay attention to that's new in your career?
Yeah, yeah, go ahead, Donna.
Please, Barry.
I do think the advances in technology are what's exciting to me, because I do think they enable a whole new world of potential for drugs and drug delivery. And I will say, and it's not a plug for Regeneron, it's really more of the reason I work at Regeneron is because we are an organization that's really dedicated to science. And so I think those of us that enter the world of drug development have an interest in science. And if you can, work through that to actually provide something to patients in need, that's really sort of the ethos of our company. And that's what I think drives all the people at Regeneron. I can say when I talk to my team, there's this common purpose of serving the greater good that, it's almost like they're drinking some Kool-Aid, but it really isn't. It's this purpose behind what we do, and putting people together that have an interest in science to be able to use that to do this.
Right. My answer is very, very similar. It is such a privilege to be in a company and in a position where you feel you have the opportunity to really touch patients' lives and make a meaningful difference. And when I started working at Regeneron, we were very focused on what is now ILEA. And my children were a lot younger then than they are now. And I still remember when one of my kids told another person that, yeah, my mom helped cure blindness, right? And that made me incredibly proud, that it was not quite true, but it does reverse one of the leading causes of blindness for the aging population. And being able to save lives, with our treatment for Ebola, and potentially helping out in a pandemic. Oh, yes, and we have a big IO portfolio. Many of us have personal experiences with friends or family members suffering from cancer, or the horrific side effects from chemotherapy. We may have lost loved ones. And knowing that we have so many clinical trials going on that may offer these patients either a cure or at least an extension of their life, that keeps me up. That's what keeps me going every single day. And although we developed the manufacturing process, I'm just keenly aware that that's what we're doing. And I'm also keenly aware that when we develop a manufacturing process, decisions we make on a daily basis could affect the safety of the drug, and that's a responsibility we take very, very seriously.
And
I think I'm starry-eyed as I was when I started.
Maybe more.
To the new people that are joining, new to their careers, how are they different, or maybe they aren't, from what you've seen years before or when you joined? What are they like today? What are they like today? What's different about them?
I don't know that they're that different. I don't know that they're that different. And I think there's a level of natural selection as well, right? So each company has a culture and an ethos. And I think, I've been at Regeneron for 16 years, and I think that is very intact. And for some people, it's exciting and they stay. And for some people, it's not, and they will leave. So there's a level of natural selection in that way. And the ethos really is that we are deeply, deeply committed to science, and there's constantly more work than we know how to get it done, but we somehow figure it out. And those are the people we're looking for. So there's a natural selection, and through that, I don't think we have a sample size that necessarily can tell you that people are different, because we select for what we know work.
Yeah, I will just say, because my group, while a lot of my group do travel around the globe, they haven't been doing that, and we've been working virtually. We don't need to be in the office in the same way that the manufacturing and lab support does. But interestingly, we kept our intern program this year, and we brought several people in who left the experience saying, I'm so interested, maybe not in Regeneron, but in science, and I'm changing my major, and this is so interesting in terms of drug development. And I don't see a difference really in the energy and excitement around science and drug development. And I think, obviously being in the middle of a pandemic, there's more of a knowledge of clinical trials than ever, probably. So this is, very ripe for those who are knowledge thirsty. But I do think that the change I do see is that people who are coming into the industry have a bit more technical knowledge. They've grown up with computers, where I don't want to speak to anyone's ages, but I did not, and I don't, that wasn't that, those were things I had to learn downstream. And so they come in and they're the people, just like my kids, that I look to, tell me how to use a new program, or they come in and they, can whiz through certain things, from a data perspective that are different. And so I think that's the difference I see. And I'm trying to figure out how do we play on those strengths and really, bring those individuals and utilize the newfound knowledge that they have that we didn't grow up with.
Yeah, it's an interesting world, and there's always something new, whether it's data or you have headsets and genetics now. It's been a wonderful conversation with both of you. I've learned a lot. I just enjoyed this very much, and I'm sure everyone in the audience has as well. So everyone in the audience, I don't know how we will do better than this, but next week we will have another fireside chat. You can join us again. You can go to IndieBio. I better read this to get this right. IndieBio.co/events. Sign up for the newsletter and come back. Thank you very much, Hannah and Bari. We really appreciate and enjoyed this conversation.
Thank you.
Thank you for the invitation. It was fun.
Great.
Have a good night. Yeah.
You too.